Hello, my name is Chris Matavi, and today we're going to be exploring the intersectionality of cellular genetics and intracranial aneurysms. So, what are intracranial aneurysms? Intracranial aneurysms, or IAs, are balloon-like structures that form in a weakened section of a cerebral artery. Over time, the vessel wall becomes thinner and more prone to rupture, leading to a subarachnoid hemorrhage or life-threatening stroke. From a cell biology standpoint, this weakening begins when the vascular smooth muscle cells and endothelial cells, which line and support the artery, lose their ability to maintain the extracellular matrix structure and respond properly to hemodynamic stress. The process starts with endothelial dysfunction. Constant blood flow exerts shear stress on endothelial cells, which normally sense and adapt to this through mechanosensitive pathways. For example, the SOX17 gene helps regulate endothelial maintenance and vascular remodeling. When genes like this are altered, the endothelial cells lose their organized structure. This makes it easier for inflammatory cells like macrophages to infiltrate the vessel wall. These immune cells release matrix metalloproteinases, enzymes that degrade collagen and elastin. If you think of collagen as like the rebar support of a concrete wall, these enzymes essentially cut those supports off. At the same time, vascular smooth muscle cells begin to shift from a contractile phenotype to a synthetic phenotype, meaning instead of maintaining vessel tension, they start producing inflammatory cytokines and ECM degrading proteins. This phenotypic switch is a hallmark of aneurysm development. Together, these cellular events, endothelial dysfunction, inflammation, and ECM degradation, weaken the arterial wall until a bulge forms. An article published in 2008 by the American Heart Association was the first genome-wide linkage study using 192 families with multiple affected family members. From this, they identified four chromosomal regions being linked to aneurysm susceptibility. Interestingly enough, the study found that smoking amplified the genetic risk on chromosomes 4, 7, and 12, a clear example of a gene-environment interaction. In other words, having the genetic susceptibility alone wasn't enough. Environmental stress triggered the cellular pathways that led to aneurysm formation. Again, in 2013, another study expanded on this work through a meta-analysis of over 116,000 individuals, identifying specific single nucleotide polymorphisms. Mutations or variants in these genes disrupt the structural proteins and regulatory signals that maintain vessel wall integrity at the cellular level. It's like removing the scaffolding and the repair crew at the same time. In 2021, a study synthesized results from numerous genome-wide association studies to identify 19 confirmed risk loci. They showed that the implicated genes cluster into three major cellular pathways. First being endothelial cell integrity in response to flow stress, second being vascular smooth muscle function and ECM remodeling, and the third being inflammation and oxidative stress regulation. Each pathway represents a point where genetic risk translates directly into cellular dysfunction. While common SNPs explain broad population risk, rare high impact variants often underlie familial aneurysms. Using linkage analysis combined with whole genome sequencing in three large dust families, a study in 2022 identified six damaging variants, most notably the FMNL2 and the TBC1D2. The FMNL2 encodes a foramen involved in actin cytoskeleton organization, which is crucial for endothelial stability and smooth muscle contractility. TBC1D2 regulates Rho GTPase independent cell adhesion, vital for endothelial junction maintenance. These genes reveal that cytoskeletal and adhesion pathways are as critical as ECM composition and aneurysm pathophysiology. Disruption of these cellular structures weakens intercellular communication and the vessel's mechanical resistance. The convergence of these studies highlights a unifying model: genetic susceptibility to alter gene expression, cellular dysfunction, and eventual structural vessel failure. At the molecular level, mutations in transcriptional regulators, such as SOX17, and non-coding RNAs, dysregulate endothelial repair. At the cellular level, smooth muscle cells lose contractility and adopt inflammatory phenotypes. At the tissue level, MMP-mediated degradation of collagen and elastin leads to vessel wall thinning and aneurysm formation. An article published in 2023 from the Brain Sciences Journal Review emphasized that these interconnected layers demonstrate how cell biology is the medium through which genetic information manifests as vascular disease. Currently, this field of study is moving toward integrating multiomics, cell culture models, and gene editing approaches to map aneurysm biology with precision. Single-cell RNA sequences can identify how specific mutations alter endothelial and smooth muscle transcriptomes. CRISPR gene editing allows direct testing of IEA-associated variants in vascular cell lines, and computational modeling of shear stress and flow dynamics can help simulate how genetic vulnerabilities translate into structural failure. Moreover, genetic screening for variants such as XOX17 and FMNL2 could help identify at-risk individuals, allowing preventative management before aneurysm rupture occurs. These are my references, and thank you for listening.